This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1223, recorded on the 30th of May, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 73 degrees Fahrenheit, which is 22 degrees Celsius, and mostly just puffy white clouds and light blue sky. Pretty nice.
Here in Chelsea, it's 23. So if it were for 28, it would be 82. So 23 is like high 70s, I'm guessing.
Right.
But I don't want to convert. It's just for other people. I want to get used to I'm working on getting used to it for 10 years now or more. 23C and mostly cloudy in Chelsea. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is also 23C here, which my phone tells me is 74.
Okay.
And it is pretty sunny out there right at the moment. I think it's going to get a little cloudier later.
Yeah, I think here at 6 p.m. it's supposed to rain. If you enjoy these programs, now this today's you haven't heard yet, but hopefully you've listened to other ones. If you like them, where we talk about science and some peripheral things that we get caught up with, we'd love your support. We give you science, we give you truth, we give you science results. We don't give you lies or misinformation, which is prevalent today in many circles, as you know. My pick has to do with that, actually. How about writing a paper and half of the references are non-existent? How do you think that would go over at Journal of Virology?
So do you think that was done with generative AI?
Hey, you know, it could have been.
Because generative AI hallucinates.
Yeah, they do that
references.
all the time. Yeah, they do. Well, they fixed it, right? But anyway, we give you the references. They're in the show notes. And we'd love your support to continue. We have 9 different podcasts here at Microbe TV in all different subject areas. So go to microbe.tv/contribute to help us out. We'd really appreciate your support. Kathy Spindler, how is ASV 2025 going?
Well, as we know, it's going to be in Montreal, July 14th to 17th. The website is asv.org/ASV 2025. So I want to point out that the timing of the events has been changed from previous years. So if you're a longtime ASV attendee, you might not have picked up on the fact that on the first day, which is Monday, in the morning are the satellites. In the afternoon, the satellites are not existent. So that's different from previous years. And in fact, it goes right to the opening reception, which is kind of right after lunch, and then the keynote address, and then the first plenary session of plenary speakers. And then there will be the first workshop with everybody giving their 12-minute or shorter talks and so forth. So that is really different, that on that first day, it's just about everything you can possibly have, from the keynote address to plenary session to workshop sessions and opening reception, and before that, satellites. So
yeah, when I was booking, I also noticed how different the last day was.
Yes.
The last day goes until the end of the day. It's not that it ends sort of at noon. And so you should be aware of that when you're thinking about your travel plans and your hotel booking plans.
Right. And if you want to stay for the party, then that's that last day's night, Thursday night. Right.
TWiV is on Thursday as well.
Correct. And I was going to say, even if you've already registered, you can add a satellite, which, as you can see, are now just in the mornings. If you see that there's one that fits your interests and your schedule, you could add that. And the early bird registration has ended, but you can still register ahead of the meeting and save money over if you register when you get there. It's going to cost you even a little bit more. So get on it if you haven't done that yet.
Is this your first year not being treasurer or secretary, whatever you were?
Yes, both. Secretary, treasurer.
So you can enjoy the meeting more, right?
I hope so. Yeah. Just go and have fun. One other thing about ASV is that there's a reservation deadline for the residence halls on campus, and it's been extended to June 12th. So if you want inexpensive housing, that's pretty nice and convenient. Go ahead and register for that. Add it to your registration for the meeting itself.
Okay, a couple of news items today before we get into our literature. The first is, this article has been everywhere. I picked one in Medical Express by Andrew Zinn, Marisha Goldhammer and Bill McCarthy, edited by Andrew Zinnin. It's entitled Make America Healthy Again Report, updated to remove non-existent studies. So this is a MAGA report purportedly written by RFK Jr. and his team. And was released on May 22nd. And some of the studies listed don't exist. And the press secretary, of course, just downplayed it as formatting issues, of course. Does not negate the substance of the report. Really. If you are using references that don't exist, it seems to me it would. For example, Noah Kreski, a Columbia University researcher, listed as an author of a paper on adolescent anxiety and depression, told AFP the paper is not one of our studies and doesn't appear to be a study that exists at all. And so, Brianne, you were suggesting that maybe it was written by AI.
Yeah,
right?
I mean, if you have generative AI write something with citations, it just makes up citations. It picks authors from some places and a journal name and a title and sort of makes a Franken citation.
Yeah, I noticed that for me, I did some kind of search on myself and it listed all these papers I'd never written. Just made them up. It's crazy stuff. Anyway, so they go through in this article, they contacted all the investigators who supposedly were cited, and they said, no, this paper doesn't exist. And the Democratic National Committee said the report is rife with misinformation. So check that out. You know, he makes a lot of claims that just do not have any substance. For example, recently he said the NIH may start its own journal.
I saw that.
Because he says the big journals are all conflicted. Okay, so where's the data that it's conflicted? Is that something you feel, or do you have data? And he didn't present any data, right? So he said they're all being paid by Big Pharma to publish their papers. Well, actually, page charges are a thing for everybody, not Big Pharma, right?
Right.
I don't know what he's talking about there. This is just crazy stuff. Okay, and that's one thing. The other one is a nice article by Carl Zimmer in the New York Times. I'm sorry, the New York Times, you know, it's paywalled. But it's called A Fungus Devastated North American Bats. A New Species Could Deliver a Killer Blow. It was published on the 28th of May. And you remember at the beginning of the 2000s that bats dying off in New York State were found to have a fungus, a fungal infection and causing a disease called white nose disease. And it was called by Pseudogymnoascus destructans. And since has gone to 40 states and 9 Canadian provinces. So, you know, they're starting to recover, it seems, but actually there are 2 species of P. destructans, and only one has reached North America. So now people are worried that the other one is going to come here and cause a problem. So you could read this article and learn all about it if you can get into the article, that is, but you could probably find something.
Yeah, I think
somewhere else.
I think it was actually 2 papers that were in the most recent issue of Science that
Mhm.
reported on this.
Right. All right, so that's our news for today. You know, we got 50% depressing news and 50%, actually it's 100% depressing.
Yeah, I was going to say, which one is the not depressing one?
Yeah, and you're right. I thought the fungus one would be a little uplifting, but in fact it's not. But now we have some science for you. We have 2 papers, and the first one, let me make sure I get the first one right, because I have started to do the main paper as a snippet, and so that's not good. But the snippet is Nature Communications, Surveillance of Avian Influenza Through Bird Guano in Remote Regions of the Global South to Uncover Transmission Dynamics. And this, let Kathy do the authors, if you want.
Okay, yeah. This is an open access paper, so you can check out the full list of authors They are from all over the world, mainly from Thailand, but it includes, I'll just name the countries: Yamagata, Japan, well, cities and countries or areas and countries, Colombo, Sri Lanka, Sheffield, UK, Sendai, Japan, Rangsit, Thailand, Brisbane, Australia, Bangkok, Thailand, Manila in the Philippines, Tochigi in Japan, Cambridge, UK, Santa Monica, California, Kulipitia, Sri Lanka, Nathaburi, Thailand, Songkhla, Thailand, Nakhon Pathom, Thailand, Kitasato in Japan, Chongqing, China, Vellore, India, Nagasaki in Japan, Cologne, Germany, Netherlands, Western Australia, Bentley, Western Australia, Concepcion, Chile, Santiago, Chile, Linköping, Sweden, Auckland, New Zealand, Toronto, Canada, Papua New Guinea, Tokyo, Japan, and another one. It says Parichat Hong Sing. Anyway, oh no, I'm sorry. That's a name. There were the second through fifth authors contributed equally, and two other authors supervised the work: Hiroshi Hamamoto and Suichi Abe. And that last name is the senior author on the paper. So it's a zillion names. And as Vincent indicated, surveillance of avian influenza through bird guano in remote regions of the global south, which includes a lot of those countries and areas that you heard me read.
Did we mention the name of the first author?
Oh, sorry. The first author is also way back in the middle of the paper, and it is Damika Wanagama. And they're from a number of those places, including Yamagata, Japan is the first one listed. So many authors, many countries, and they collected bird guano from the global south, which doesn't include South America, but includes a lot of the other countries in the global south, to survey avian influenza. And they found H5N1 avian influenza was the most prevalent. They surveyed islands, remote rural areas. Some of them are affected by wars, instability. In other words, they're in conflict zones. Some of the areas have rare earth metals. So some of this plays into thinking about why we want to do surveillance or be aware of what's going on in those regions. And they also looked at prevalence of these avian influenzas with respect to bird migratory routes. They collected over 27,000 samples from a period from December 2021 to February of 2023. So a little over 2 years and 27,000 samples. And they evaluated for influenza M gene by RT-PCR and then by sequencing for hemagglutinin and neuraminidase. They evaluated times at which the avian influenza was prevalent, and there was variability in that. 8 out of the 10 countries had their highest avian influenza RNA in December to February of 22 and 23, which would be in the summertime. So I found that interesting. And not only was H5N1 dominant, but it was dominant across multiple regions. That's my summary. That's what I got from it. And then I'll leave it to the others to tell you more of the details.
All right. So as everyone should know, if you're listening to TWiV, the avian influenza, pathogenic avian influenza viruses, highly pathogenic avian influenza viruses, HPHI, outbreaks have increased among wild birds, and there is increasing crossover into mammals and humans who are also mammals.
Yes. And also, domestic birds, farmed, commercial, poultry.
Right. And the point of this paper is that in low and middle income countries, surveillance programs are scarce, and that may be where a lot of the crossover occurs, as you see. So that's the purpose of this paper, this work. From December 2021 to February 2023, they did a surveillance study. They collected fresh bird guano from remote uninhabited regions of 10 countries: Indonesia, Madagascar, Maldives, Mozambique, Myanmar, Papua New Guinea, the Philippines, Somalia, Sri Lanka, and Yemen. Had to look up where the Maldives were. It's the only one I didn't know. And so this is giving a new viewpoint on where these viruses are. And there's not just H5N1. As you'll see, there are many other types of subtypes of influenza viruses. So they had 27,036 bird guano samples, which they assayed for viral RNA, and 360 had RNA in them, 1.33%. And there were lots of variations. So there were geographic variations in the percent positive, that is. So in Myanmar, 0.9 to 1.7%, Papua New Guinea under 1%, Indonesia 0.4%, just to give you an idea of what the percentages were. Philippines 1.7. The highest percentage were found in Eastern Africa and the Arabian Peninsula. And they have lovely maps where they put all these data on, so you can see where the Maldives are.
Yes.
The other M country I didn't know was Moldova, so it has nothing to do with this study, but it came into my head, and it's a former Soviet republic, which Russia doesn't care about because it's landlocked. It's right next to Ukraine. I think it's between Ukraine and Romania, right?
I believe so, yeah.
And they don't want it because there's no coastal access and there's no precious metals and all that stuff. Anyway, those were the highest in Eastern Africa and the Arabian Peninsula. Madagascar, in one of the islands, 2.59% positivity. Sri Lanka and the Maldives, 1% to 2.3%. So these study areas, by the way, they give us some information. They're all remote, rural locations. They're often uninhabited islands. I'd like to go to one of those. Small fishing communities. There's not going to be any cell phone service. Indigenous tribal settlements that rely on hunting and gathering. No history of poultry farming. Okay, that's important. So we're talking all about wild birds here. And as Kathy pointed out, some of these regions are affected by conflict and instability, which, of course, not only is a problem for health care, but also routine disease surveillance. Yeah, so those are the percentages. Yeah, go ahead.
I was going to say, I'm glad that they mentioned that, because when I was reading the earlier parts of the paragraph, I started to think about, are some of these places, in Eastern Africa and in the Arabian Peninsula, more densely populated, potentially with animals or people? Or is that why we might see such big differences in these percentages? And so it was good to sort of have them point out, no, these are all kind of more rural locations. And so this is not necessarily based on things like poultry farming or, dense populations in those areas.
Yeah, right. All right. So besides percent positivity, how about spatiotemporal patterns? So where in the world and during what part of the year? And so there was quite a bit of variation. For example, from December to February is low compared to April to June, August, October, December to February. So in December to February 2023, so the first one was 2021, it rose sharply there. And then you have a lot of difference among countries. I'm not going to go through all the list because they have every country with the p-value showing how you have heterogeneity, but there was country level spatial heterogeneity as well. Temporal shifts also between seasons, just as an example, because there are a lot of examples cited here. Between December and February 2021 and 2022, and April, June 2022, Madagascar, Yemen, and the Philippines had the largest increases in percent positivity. And they go through some other countries, but you get the point here is that you can see differences in when it's happening and in what countries.
It seems like overall there's a general increase over time.
Yeah, which is not surprising as these things are circulating more and more every year. 8 of 10 countries reached their highest mean RNA concentrations during December, February 2022 and 2023. So I don't need to tell you the countries, but 8 of the 10 that they studied. And across seasons, there's a lot of variability in viral dynamics. Some countries show high seasonal fluctuations, others less. Okay, so then what's the most frequently detected subtype? As Kathy told you, H5N1, with the highest detection in Gan and which is in the Maldives. 85.7% of the samples were H5N1. I love the names. We have Gan and Pigeon Island and Makandudu, which is in the Maldives as well. H1N1, peak prevalence, so it's another highly pathogenic avian influenza virus. Peak prevalence in Papua New Guinea, 50%, H2, and they go through all of the subtypes. I'm not going to go through all of them, but H2N7, most frequently detected in Indonesia, H3N6, H4N6, H5N3, et cetera, et cetera. Okay, so Papua New Guinea has the highest subtype diversity of all the countries, 0.39 subtypes per positive sample, followed by Indonesia and the Philippines, whereas Yemen and Somalia had lower diversity. So that's, a lower diversity may mean a stable ecology where not much is changing. That's what they suggest later. And then they have some sequences, of course, because they're doing PCR, so they do phylogenetic trees. And you can see distinct clustering pattern for both the hemagglutinin and neuraminidase genes, which are the ones they got. They don't have the full genome here. And these phylogenetic trees show that the samples cluster with other samples that have been taken from Asia, Middle East, Europe, Africa, North and South America. And what I think is cool is the clustering patterns align with major bird migration pathways. And in fact, sequences from a number of countries overlap with key bird migration routes. And by the way, I meant to tell you those. I think it's very cool that there are very specific bird migration routes, like there are airplane routes, whereas the East Asian Australasian flyway, the Asian East African flyway, the Central Asian flyway, the Mediterranean and Black Sea flyway, and the East Atlantic Americas flyway. And that's shown in one of the figures, actually. Okay, so the scrolling back to that. So a lot of the diversity, the clustering of the HA and NA genes, is aligned with these bird migration routes. Also, H5N1 was dominant across multiple regions. They do detect other subtypes, but in Maldives, Sri Lanka, Madagascar, Somalia, Yemen, Mozambique, Philippines, Indonesia, and Papua New Guinea, H5N1 was dominant. And the majority were classified under clade 2.3.4.4b, which is the one in cows in the U.S. currently. And other birds have that clade in Asia, Middle East, Europe, Africa, North and South America. And they say, interestingly, samples from the Maldives, Sri Lanka, Somalia, Yemen, and Papua New Guinea in 2021, 22, and 23 show a close relationship to viruses currently circulating in dairy cattle. So these viruses move around because birds move around, right? Incredible. By those flyways, they have it all mapped out. Have great radar. And also, they note that in some places, they find highly pathogenic avian influenza virus sequences, but there's no reports of disease in birds caused by these viruses. For example, they say we couldn't find any specific reports from the UN and the WHO about avian influenza in Sri Lanka, Myanmar, Papua New Guinea, Mozambique, Madagascar, Yemen, Somalia, and the Maldives, which were all places where they found sequences, right? In the Philippines, though, that's not the case. They have had H5N2 outbreaks, and they did find H5N2 actually circulating a couple of years before the outbreak. So it's been there for a while. And they also say that these H5N1 positive samples were from Somalia, Yemen, and the Maldives have a amino acid change, H275Y, in the neuraminidase, which is a known substitution associated with resistance to neuraminidase inhibitors like Tamiflu. So, I mean, these viruses are out there circulating. They've never been exposed to Tamiflu, and they have resistance because that's how it happens. It just randomly arises.
Do we think that the reason why they haven't seen any, or they couldn't find prior reports about disease may be due to, not great surveillance for disease? Or maybe there was disease that was missed and this is in some ways more sensitive?
I'm sure that that's a possibility, right? And these countries don't have great surveillance. Papua New Guinea, probably not, right? They also looked at the cleavage sites in the HA. So the HA protein has to be cut. A lot of people don't understand cleavage. It's very interesting. What does that mean? Well, it means it's cut with like a pair of scissors. So that's why I say cut by an enzyme called a protease to be infectious, right? And so they look at all these sequences around the cleavage site for all these different subtypes that they see. They just list them, right? They don't say anything about them. But I know that some of these viruses have a furin cleavage site. So human influenza viruses have a cleavage site that is cut by proteases that are restricted to the lung. All right. And so that's a major determinant of the tropism. The viruses don't reproduce in other tissues because the proteases aren't there. But many, especially H5N1, I know of, HA has a furin cleavage site, which furins are everywhere in all tissues. And so it gives it the ability to reproduce in other tissues. And a furin cleavage site is multi-basic, 6 or 7 multi-basic residues, like lysine and arginine. You remember your basic amino acid residues? Probably most people don't. I had to look it up. So KRRRKR, for example, that I see in H5N1, that's a multi-basic cleavage site. RRRKR, that's found in a bunch of them. So that's what I noted there, but they didn't actually say it. They mention it in the discussion, but they don't say it there. Okay, so that's the data. Those are the data. And so basically we have new information about sites that haven't been sampled very much for avian influenza viruses. The discussion actually has a number of really good talking points here. Let's go through just a few of them. So first, they have a high richness of subtypes in Papua New Guinea, Indonesia, and the Philippines. They think that maybe this suggests a more heterogeneous population driven by maybe ecological niches, host species, or transmission interfaces. But of course, when you have different subtypes circulating, they can reassort and make new viruses. So that's the concern there.
Right. And you could also imagine, as they say, kind of those are areas where there's a lot of biodiversity. So if there are lots of different birds, then you could imagine a lot more sharing of viruses and a lot more viral mutation to adapt to the various bird species.
And conversely, they don't have a lot of subtypes in Yemen and Somalia, so maybe things are more ecologically stable or transmission-wise stable there. So several, this is my favorite part. Several high-risk wild bird species contribute to avian influenza transmission across these countries. So waterfowl, including the lesser whistling duck. It's in Myanmar, Indonesia, Philippines, etc.
Is there something called the greater whistling duck? Checking.
Check it. The northern pintail, which are in all the countries they sampled. The gargany, the Pacific black duck, are known carriers of H5N1 and H9N2. And here's the key. They frequently mix with domestic ducks in flooded rice fields and wetlands. especially in rural farming communities. So they go and they chat. They have a cafe clatch. Is that the word, cafe clatch?
It is. It is.
I like this idea that birds are getting together, but they share their viruses then, right?
They have there are many whistling ducks, but I don't see one that's greater. They're just
Maybe it's just whistling duck and the lesser whistling duck.
No, there's all sorts of, there's like wandering whistling duck, black-bellied whistling duck, fulvous whistling duck, but no greater.
No lesser.
There is lesser though.
I'm sorry, there's lesser, no greater. Yeah, okay.
Yeah. I just feel bad for the lesser duck.
Well, the lesser duck goes to these flooded rice fields and hangs out with domestic ducks. And then we have the common sandpiper, the great knot, the whimbrel. These are birds that follow major flyways like the ones I mentioned just earlier. And they stop, it's like you're on the turnpike. They stop at key wetlands, rice paddies, and coastal lagoons in the countries where they're found, where they can transmit their viruses to local poultry and wild bird populations. It's just the mobility of birds is the key here. It's really remarkable. Gulls and terns, the black-headed gull, the whiskered tern, they frequently scavenge in fish markets. You've seen seagulls scavenging at inland markets, right? And you go, hey, we're pretty far from the ocean here. They're hungry. They frequently scavenge at fish markets and coastal areas in low-income fishing communities, and again, increasing spillover risk between wild birds, poultry, and humans. I'm going on with this because I really love these birds. Herons, egrets, and ibises or ibises. How do you?
I think it's ibis.
Ibis, such as the little egret. There's another little lesser, and the glossy ibis inhabit rice paddies, irrigation canals, and marshlands, and they mix with wild and domestic bird populations. Then we have our birds of prey, the Brahminy kite, the eastern marsh harrier. They scavenge infected bird carcasses, right? So they get infected that way. And then we have urban adapted species, right? There's always those. The common myna, the Eurasian tree sparrow, and the house crow. I love that. House crow. And you can tell the difference between a crow and a raven in the way they caw. I was looking that up the other day because there was a big black bird flying and making noise. I wanted to figure out what it was. I think it was a raven. Nevermore. And they spread the virus in small village communities, informal markets, and backyard poultry farms. Backyard poultry farm, you have poultry waste, open food sources in the markets, live bird trade. So lots of cross-species infection. So this was to me eye-opening because there are lots of specific ways that different species can spread these infections. They also talk about how the differences in the RNA concentrations that we have seen in the paper probably reflect migration patterns, environmental conditions, and the bird community composition. And they notice that there's a biphasic pattern of RNA levels, 2 periods of heightened viral activity. One is April to June, the other is December to February. And they say these are probably corresponding to seasonal triggers like rainfall, food availability, the congregation of migratory birds, which happens in tropical and subtropical ecosystems. And so they say, if you do surveillance, you shouldn't have it based on temperate climates, right? You have to use the seasonal frameworks to do proper surveillance. Because if you're going to look in times when there's nothing there or very little, it's not useful.
And these might be better places to look because they don't have the same kind of temperature fluctuations given how close they are to the equator.
Right. They also point out that these increases in concentrations in some countries in the months like December, February 21, 2022, and followed by declines point toward episodic amplification events rather than continuous circulation. So that's one of the major take-home points here. There isn't continuous circulation. They're episodic infections and transmission. So they think these are driven by ecological pulses like migration or breeding events rather than persistent endemicity. They also comment on the spatial heterogeneity, which they see. Some countries have divergent patterns of increase and decline across seasons. They say this probably reflects localized risk factors like the kind of habitat, the species diversity, climate, and proximity to migratory flyways. So countries along the key migratory flyways or at ecological convergence zones have higher variability and sharper seasonal peaks. And again, they say, so the transmission is dynamic and nonlinear, so you have to adapt your sampling to that. They also point out that the RNA concentrations were the highest during December, February 22 to 23, higher than the previous year. And this occurred before the widespread surge in avian influenza that started in 2023. And that has what has led to infections across multiple species in multiple continents, including cows in the U.S. and some humans as well. So again, this this peak occurred before we had reporting of disease. And so they say, they also quote the Philippines example again, the virus was present in the region much earlier, and they say this is a problem with surveillance. And they finally make a sentence about the presence of polybasic motifs in several sequences raises concern regarding the potential emergence of highly pathogenic strains. That is because they would reproduce in multiple organs, not just in the lung. They also say this amino acid change in the neuraminidase of some of these samples raised concern about antiviral resistance. And the places where they got these samples are along the major flyways, so those changes could be transmitted to other parts of the world. Also interesting is there is a lot of mining, logging, and infrastructure projects in Indonesia, Papua New Guinea, Madagascar, and Mozambique that are pushing human settlements into remote forested regions and coastal islands, even areas that are previously dominated by wild birds and migratory waterfowl. So these are just ecological changes that are potentially driving some transmission.
Yeah, they're also making people be a lot closer to those birds.
Exactly. And they say also the increasing demand for nickel, cobalt, and rare earth metals for batteries, right, has led to deforestation, wetlands destruction, and the creation of artificial reservoirs and open pit mines which attract birds. And the workers are near these industrial sites. So as Brienne says, human-wildlife contact increases. As for conflict, there's conflict in Myanmar, Somalia, and Yemen. And this makes HPAI worse, potentially. Civil unrest, displacement, food insecurity have led to unregulated poultry trade, overcrowded refugee settlements, weakened veterinary and public health infrastructure, and this all accelerates the risk of transmission among humans, poultry, and wild birds. And they say during these breakdowns in war-torn areas, many people hunt wild birds or consume poorly regulated poultry. So that's a problem. And many migratory birds stop in these conflict zones, like the northern pintail and the whimbrel and the black-headed gull. So that's the story here, that they wanted to basically show the inequities in global infectious disease surveillance. We currently do surveillance in developed regions or areas with large-scale poultry farms. So there's a lot of areas of zoonotic potential that are not sampled or not under monitor. So they say no region, and also they say we're not trying to blame anyone in this paper. No region should be framed as solely responsible for the outbreaks. We don't want to blame anyone. We just want to emphasize the value of good surveillance. We got a paper on TWiV, an email on TWiV last week. And someone said, you shouldn't say the Chinese vaccine. That's not necessary. And they're right. Just say Covaxin or whatever the name of the vaccine is. And so basically here you have some sequences derived from an unusual kind of surveillance, and this says we have to do this more. And these areas that have been surveyed are dynamic areas, so you have to do surveillance frequently.
Sort of also, reminds us that viruses are everywhere and do not stay in one place. And so we need to care about the viruses in lots of parts of the world. And we need to care about what's going on in lots of parts of the world if we're going to maintain our health.
Right. And I keep reading this with worry in my mind because, we left the WHO. We're not helping anyone anymore. And so it's bad. It's very disturbing to me and to everyone else, I'm sure. All right. So that was very cool. I like that, particularly the birds.
Although I also didn't know it was called bird guano. I thought
You always saw bat guano, right?
yeah, I thought guano was specific for bats.
Yeah.
So that was the first thing I learned when I read this paper.
It's not, right? It's birds
Yeah.
do? Okay. Our second paper, ACE2, angiotensin converting enzyme 2. It's a human protein. Well, it's multi-mammal species protein, a host protein present on multiple cells, which serves as receptors for various viruses. ACE2 from Pipistrellus abramus. Bats is a receptor for HKU5 coronaviruses. Kathy, you want to do us the honors?
Sure. There are co-first authors, Nicholas Catanzaro and Xian Wu, and the senior author is Michael Letko from Washington State. And then there are investigators from UNC at Chapel Hill and Caltech, and as well as Washington State. And so the short summary, give you a little background. The three main coronaviruses that emerged in the 21st century are SARS-CoV, also known as SARS-1, MERS, and SARS-2. So the subject of this paper is the MERS viruses, and those were first found to be transmitted from camels to humans, and so the ME of MERS comes from Middle East, where it was first identified. And so the MERS-CoV causes a severe respiratory disease. And virus discovery has identified hundreds of viruses from this MERS-CoV family, and they're called merbecoviruses. They're a subgenus of the beta coronaviruses, from which we get the BE and the CO, the Beco, and the MER being from the MERS. So we're going to be talking about merbecoviruses here, as contrasted with SARS-becoviruses, which are the SARS-related beta coronaviruses. So there's lots of these that have been identified, but few of them have been studied. And so the big question is, what are the entry receptors for these many merbecoviruses? And so they use an approach that the senior author, Michael Letko, developed, it looks like when he was in Vincent Munster's lab at Rocky Mountain Labs in Montana for SARS. And so he's now applied that with this group to work on identifying entry receptors for the merbecoviruses. So to review, the primary receptor that the MERS coronavirus uses is called DPP4. And then SARS-1 and SARS-2 use ACE2, the one that's in the title of this paper. Some other MERS viruses also use ACE2, and we're going to find out more about that in this paper. And so they're asking the question, what about this large collection of the merbecoviruses that are out there? What do they all use? And so they've named this approach merbecotype, modeling it after the name that they used when they did this for the SARS viruses. They called that sarbecotype. And it's a pseudotype approach, and I think that's where we get the type of the name of this approach. And most of the merbeco sequences are just that. They're sequences. They don't have viruses that those sequences come from. In other words, so we have a lot of sequences, but we don't have the viruses. So how are we going to identify the receptors in the absence of having the viruses? So they engineer vesicular stomatitis virus, also known as VSV, which is a negative strand RNA virus. And they engineer it so that it can have envelope proteins, surface proteins, that basically consist of a foreign spike protein. And here they're using it from the merbecoviruses. And so when they make one of these viruses, it's called a pseudotype. And here it's called a MERS-CoV to be exact. And so these pseudotype viruses, in addition to having this a MERS-CoV surface spike protein on them, they have a reporter gene, luciferase. And that reporter gene is what they use in their assays to measure how well their pseudotype viruses are going to enter into cells. So it's just a nice reporter assay. And so what's the source of the sequences, that they use to make these pseudotypes? Well, they go to GenBank and they go through this whittling down process, and they end up with 65 MERS-CoV virus spikes. And those 65 spike sequences have 34 unique receptor binding domain, or RBD sequences. So these came from 18 years of virus discovery in 6 countries and 3 continents. And they point out that some of this virus discovery preceded the identification of the MERS coronavirus that started all of this. And so they spend a fair amount of time and description of how they developed the pseudotype vectors for these MERS-CoV viruses. They couldn't just do it this kind of the same way they did for the SARS ones previously, but they did that very carefully, and they optimized them for their entry assays. So this actually represents a ton of work. And they start with cell lines that don't endogenously express either the ACE2 or the DPP4, and then they're going to introduce those receptors and a third one into those types of cells. And then they can use those specifically engineered cell lines for their assays with these pseudotype viruses. And so they talk about the fact that there are 4 clades of the MERS-CoV viruses, so 4 groups of their based on the receptor binding domains. And those are defined on the basis of sequence, insertions and deletions, which they call indels, and their entry phenotype, which is kind of what they're characterizing in this paper. And the geographic location of origin of these MERS-CoV viruses is not a defining factor for the clades. So the key finding of the paper is what's indicated in the title, that one of these clades, which has all of the HKU5 MERS coronaviruses or MERS-CoV viruses, that this clade uses ACE2 from this particular species of bats, the Pipistrellus abramus bats. And so these Pipistrellus abramus bats are also known as Japanese house bats, but that's somewhat of a misnomer because they are found across East Asia from China and Taiwan up into the Korean Peninsula and Japan. And the important thing about these, what are they called again? Japanese house bats, that house kind of gives it away, is that they're what is called synanthropic. And that means they've adapted to living in proximity to humans, and they benefit from environments that humans have modified. So they pose opportunities for human exposure because they're constantly in these environments where there are humans. They don't have, these bats don't have good compatibility with, the viruses that they identify don't have good compatibility with the human ACE2 at the moment, but they're specifically compatible with this bat Pipistrellus abramus ACE2. So there's some distinction there at the species level between the ACE2s. And so this is important from the standpoint that we should be paying attention to it because these bats are all around people, all around Asia. And so in addition to identifying ACE2 as the receptor for this clade of bats, they make an infectious clone of one of these HKU5 viruses synthetically, and they demonstrate that it uses ACE2 by a number of nice assays. So they're not just working with the sequences anymore, they actually make a virus, and they use several assays, including our favorite plaque assays of infectious virus to show that indeed they use this specific ACE2 receptor. They also do some mutagenesis and structural work at the end of the paper on the RBD and its interaction with this ACE2, which is almost in a way, to me, an add-on to the finding of the paper and could be a separate second paper. So that's my summary.
So what was the word for living around houses again?
Synanthropic.
Yeah, I never heard that one before.
Mm-mm.
Dixon always called it peridomestic.
Yeah.
That would make sense too.
Right.
Yeah.
I
In
like
fact,
that.
one of the dictionaries looked at on my phone said, "Oh, you have to upgrade to the paid version if you want this word." So I said, "Forget that," and I just
Wow.
Googled it.
What an income strategy.
Yeah.
All right. That was the short version. I'm going to do the long version, and there will be some repeat. But the short version is meant for people who just want to listen to that and then go. and
Or
cut
have
their
the
grass.
framework to hang the long version on.
Yes,
Yes.
that too.
So-
That works for sure. Sometimes in the past I have felt bad repeating, but repetition never hurts. As Peter Palazzi used to say to me,
That means your listener can feel smart.
especially
Yes.
with medical students.
Yeah.
MERS coronavirus, discovered in the Middle East, 2012, causes severe respiratory disease in humans. It's endemic in dromedary camels in this region, Arabian Peninsula, and they're all infected at birth, and they transmit it to humans. So there's always a continual number of cases every month. Not many, a couple of cases a month, because of that. Camels are very common there, right? They use them as pets. They use them to race. They sometimes eat them. So people raise lots of camels there. So they're all infected at birth. They don't get very sick. They get some runny nose and stuff like that. Maybe they feel badly, but they can't tell us. So, since then, people have said, where else are there viruses? And there are hundreds, and they infect lots of different animals, not just camels, across multiple continents. And, most of these are sequenced genomes. And now, not very few, they say, actually, majority of these have never been isolated as infectious viruses or characterized. And so we don't know if they're a threat or not, because you can't put them in cell culture and do some experiments and say that, can they bind human ACE2, which would be step one. So the entry step is the focus of this paper. And MERS coronavirus, like the other coronaviruses, have a spike protein embedded in the viral membrane. And first, there's a series of interactions that occur in order to get the virus in the cell. The first thing is the N-terminal domain, the amino-terminal domain. I know a lot of people, we say things and goes over your head. So protein has an amino and a carboxy terminal N. That's a chemical thing, NH2COOH. And so we say the N-terminal as an abbreviation. The N-terminal domain of the spike attaches to lectins, carbohydrates present on the cell surface. Then the receptor binding domain of the spike binds to the host cell receptor molecule, and it reorganizes the receptor. The spike reorganizes, we'll talk about that later, and that mediates fusion and subsequent cell entry. And often the reorganization is preceded by a proteolytic cleavage, which can happen at the cell surface or it can happen within endosomes, depending on the cell type. And each of these functions is done by a separate region of the spike. They're very compartmentalized on the spike protein. So the receptor binding domain, which is a separate domain you can make, that's Peter Hotez's vaccine that he charges $3 a person for in India. That's a receptor binding domain protein made in cells. And that's the one that has the core interaction with the host receptor. So the receptor for MERS coronavirus that infects camels and humans is dipeptidyl peptidase 4, DPP4. It's a plasma membrane protein, and it happens to be an enzyme. It's a peptidase. Cuts proteins. But they say there are other viruses that don't bind DPP4, and some of them bind ACE2.
Yeah.
to infect hosts.
And many of these are in other bat species, particularly in some of the Egyptian Rosetus bat species. And some people might sort of wonder, well, why do we care about, viruses that bind to bat ACE2 versus whatever else? And I think that it's really important to think about, this is one of the ways that we know kind of the breadth of what's out there and sort of what's possible evolutionarily. And so we can think about, okay, this is the broad set of all of the things maybe nature has tried to make.
Yeah.
And then we can say, oh, a particular virus like SARS-CoV-2 has this sort of binding site, but that's may not be unique or has a particular cleavage site that may not be unique if we look at the whole set of viruses that are in nature. That type of binding may have evolved multiple times. And so that might be something we see showing up over and over again if we actually know the full breadth of what's out there.
So the merbecovirus RBD is also a single domain that works by itself. And they've taken advantage of this previously. By the way, Michael Letko, senior author here, was previously on TWiV, as was Ralph Barrick, as was Pamela Bjorkman. Well, three of the authors have all been on TWiV. And so they have made a cassette where they can swap out the RBD of SARS-CoV, a sarbecovirus, with RBDs from other sarbecoviruses. And then you can take that hybrid spike and hybrid spike protein and put it in a VSV backbone so it's safe to work with. And then you can measure entry into cells in culture. So they call this sarbecotype because you could look at different RBDs and get an idea of what they're binding to. Right? You could do that rapidly without having to get infectious virus, right, for each one of them.
And it's also easier and cheaper than having to make just the whole spike.
Sure. You don't, yeah. So in this paper, they're making a new approach called merbecotype. It could be a name for something, merbecotype. I don't know what, but it sounds good. It's got a good ring to it. I like sarbecotype too. So basically, they can swap out RBDs of merbecoviruses. So the RBDs of these viruses are classified into 4 clades based on the presence or absence of insertions and deletions. And at least half of the merbecovirus RBD clades use ACE2 for cell entry. And there's a whole group of HKU5 viruses with different numbers that you'll see they have a preference for ACE2 from their host species, which is Pipistrellus abramus bats. Pipistrelli, for example, is the Italian word for bat. I like that very much. Because when I was a kid and I went to Italy when I was 8 years old, they were always talking about pipistrelli sono qui. The bats are here. So they look at the binding of these HKU5 spikes in this paper, and they also just determine the structure of the spike trimer, and that's interesting as well. We'll talk about that. So they want to test a lot of RBDs from a lot of different merbecos. That's the whole point here. So they make a construct where they can pop in RBD from other merbecoviruses. And the backbone is MERS coronavirus, right? Because we know a lot about that. And so they take this spike and they human codon optimize it because it's from camels, right? And camels have a different genetic code than humans. And so you want to get good protein expression. So you make sure the codon usage, because, there are multiple codons for each amino acid.
It may not be that they have a different genetic code. They may have a different abundance of tRNAs that they use for their genetic code.
A different preference for which codon is predominantly the one that makes that amino acid.
That's right. That's the correct way to say it, yeah. So they codon optimize this MERS coronavirus spike. They put a sequence at the end so they can detect it. They put restriction enzyme sites on either side of the RBD so they can pop it in and out. Restriction enzymes or enzymes that cut a specific sequence. You can buy them and use them to do genetic engineering like this. All right, so then they have this. So MERS, remember, MERS coronavirus binds DPP4, but ACE2 is also a receptor for other Merbecoviruses, including one with the the PDF virus, the portable document format virus, PDF 2180. So they took the RBD from PDF, well, I'll just call it PDF, and they showed that you could put that in to this MERS corona and show receptor switching, because the MERS coronavirus RBD binds DPP4, and if you put in the PDF RBD, now it binds ACE2. So they have to figure out where the RBD is, and they say that's not easy because there's not a lot of conservation across all these Merbecovirus sequences, so they have to do a lot of fussing around, and they notice that there are a couple of glycines which seem to flank the RBD. So they make a couple of different chimeras to maximize their chances of getting it right. And what they will do is make the spike with the RBD put in it, and then put that spike into vesicular stomatitis virus, which then you can use to infect cells safely at BSL2 level, right? As long as you don't stick it in your finger, which some people have done. Probably Kathy knows all about that, being in John Holland's lab. John Holland used to tell me about that.
Yes.
Okay, so then they say, what cells are they going to use? They decide on 293 T cells, which they say they have a low, but not complete lack of susceptibility to coronavirus entry. And then they can put into those human ACE2 or human DPP4 or whatever species that you want, and you can infect them. And so they say that if you use pseudotypes with the chimeric spikes with the PDF RBD, they can bind to ACE2, human ACE2. So they've made a successful exchange of the RBD.
It's sort of interesting to note that they mentioned using other cells and them not working. And it seems as though the cells that they need here are human origin cells.
Right. It's funny, right?
Mm-hmm.
All right. So, yeah, because they tested baby hamster kidney cells and they weren't.
Human cells make human proteins. Well, what?
They do. So they also put a RBD from HKU4-SM3A virus. And this utilizes DPP4, and that also works in the context of the MERS coronavirus spike. Okay, so the system is working, and then they want to optimize the backbone for pseudotype efficiency. You know, they want to be able to detect low efficiency viral entry. And so one of the considerations is the protease that cleaves the spike protein, and one of them is part of the TEMPRESS family of cell surface proteases, TMPRSS. And then also in the endosome, there are cathepsins, and either of those can cleave different coronavirus spikes. Furin is another protease that can play a role in entry. And MERS corona spike contains furin sites, by the way. And these furin sites, they say the removal of the furin site from SARS-CoV-2 gives increased pseudotype efficiency. It's very interesting because if you take it out of bona fide SARS-CoV-2, it reduces infectivity. But in the pseudotype, it increases it. So they took it out. They took out the furin site. They took out some C-terminal. They put in some C-terminal truncation to make it more effective based on the literature. But in their system, they didn't matter at all. Those modifications didn't matter. I probably shouldn't have told you that, but that's how science works. You read the literature, you say, oh, we should do this and this. You put that into your construct. It doesn't do anything, but you have to try it.
It shows that we don't know everything before we go into our experiments.
No. All right, so now they have, their GenBank has 2,000 entries for Merbecovirus and MERS-like viruses. 842 have the full-length spike. Half of those are from species other than humans and camels, which they don't want because we want other species. So they end up with 34 unique RBD sequences. which span 18 years of virus discovery, 6 countries and 3 continents. Sounds impressive, right?
Mm-hmm.
And they can look at these sequences and put them into the 4 clades, 1 through 4. Clade 1 includes MERS and HKU4. Clade 2 includes HKU5. Clade 3 includes the African myrbecoviruses. Neo-CoV, it's like from the movie Neo. What's the
Matrix.
The Matrix, thank you. Neo-CoV, I love that name, and PDF virus. And then clade 4 contains the hedgehog myrbecoviruses. More cool species. I love this.
Well, and they call them the ARN-CoVs.
The ARN-CoVs, yeah. So
That
anyone
comes-
named Aaron, you have a virus named after you.
It comes from the name of the host species,
The hedgehog.
Erinaceus.
Erinaceus, yeah.
Or that's the genus name. Yeah.
It's the genus for hedgehog.
Huh.
Right.
So people who have the name Aaron, you have some, allegiance to pay to hedgehogs, I guess. So they take RBDs for these Myrbecos that they've got, the sequences, they put them in their spike background, they make viruses, and they test them on 293 T cell lines transduced to make human coronavirus receptors, either aminopeptidase N, DPP4, or ACE2. Okay, so without trypsin, now trypsin is another way you can cleave the spike if you don't know what protease is that's cleaving it. And sometimes that will let you get into the cell surface more efficiently. So without trypsin, the original 293 cells are not susceptible to any of these pseudotype viruses. They don't reproduce in them. The APN producing cells can be infected with 229E, which binds to that receptor. Okay, so that's a good control. The cells that have human DPP4 are susceptible to clade 1 RBDs, but not the others. And cells producing human ACE2 had low susceptibility to SARS-CoV-2 and clade 3 myrbecoviruses. And they do experiments with trypsin that moderately increase the entry in some of these cases. So the human cells with ACE2, human ACE2, can be infected with a wide range of myrbecoviruses. So they investigate whether these their RBD panel can bind to receptors from the cognate host species, not just human, right? So ACE2 and DPP4 were made from the European hedgehogs, Erinaceus europaeus, as Kathy said, and that's the natural host species for the ARN-CoVs. And ACE2 was made from the Japanese house bats, Pipistrellus abramus, which is the natural host for the HKU5 viruses. So it turns out that DPP4 and ACE2 from the hedgehogs were inefficient as receptors and didn't allow entry for any of these myrbecovirus RBDs in the absence of trypsin. They actually take a shortcut here. They say did not mediate entry for any of the myrbecovirus clades. Well, a clade cannot enter a cell. Right, Kathy?
Right.
Yeah.
So from the RBDs from different clades, there's only a couple of extra words, it's not so bad.
For any members of the myrbecovirus
Yes,
clade.
right. So in the absence of trypsin, doesn't work with hedgehog, DPP4, and ACE2 with any of these 34 RBDs. But if you use ACE2 from the Japanese house bats, they're highly permissive for entry by the entire HKU5 cluster of clade 2 myrbecovirus RBDs with or without trypsin. So that's cool. ACE2 from the bats, these HKU5 viruses will bind to that. And they spend pretty much the rest of the paper looking at that.
So the one thing they don't really mention is, does that mean that there's some other receptor in the hedgehog? If some
Must
of these
be.
are
Yeah, there must be.
are hedgehog as the host species.
Yes, that must be, right? Because they
Right.
do infect them.
They discuss that in the discussion. They also say it could be that they just don't have the assay optimized,
Yeah.
and it could be ACE2 or DPP4.
Because they have only RBD, right? And the other
Right.
parts of the molecule could matter. Because the rest of the molecule is MERS
Right.
coronavirus spike. So they just have to make the whole one. But that for screening, they didn't want to do that, as Brienne points out. It's more expensive. Okay. So there's a little bit of infectivity in 293 cells with these RBDs. So they decide to try BHK cells, which aren't susceptible to any Sarbeco or Mabeco virus. Oops, I see a typo there. They left the O out. So what they do is they take BHK cells, baby hamster kidney, and put in Pipistrellus, ACE2, and they now become susceptible, highly permissive for HKU5 RBD-mediated entry. So that they conclude that all HKU5 sequences are coming exclusively from Pipistrellus abramus samples. So just to be sure that this is not a background thing, they do that in the BHK5 cell. So that's a real result. All right. So so far, what they have done is to put a plasmid encoding the receptor, like ACE2, into cells, and you get a transient production of protein. It's not stable for a long time. It doesn't last. The cells last forever. You can keep dividing them every week, but the protein goes away eventually. So they wanted to make cell lines that produce protein forever. So they use a lentiviral vector to deliver the ACE2 from Pipistrellus into Viro cells. And these are now, if you put Pipistrellus ACE2 into Viro cells by this lentivector, they're susceptible to the entire HKU5 RBD panel. And, there are a lot of HKU5s. There are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 different HKU5s that they have in their collection. And so all of these will work with Pipistrellus ACE2 delivered by lentiviral vector. So that's clade 3, well, that and clade 3, including neo-CoV and PDF viruses, but they're poorly susceptible to clade 1, 4, and clade 2 that were not identified in Pipistrellus bats. Makes sense, right? This is a Pipistrellus ACE2. So they wanted to make sure that this is all okay. So they take the full-length spike from one of the HKU5 or two of the HKU5. They make viral pseudotypes, and these will infect BHK cells with the Pipistrellus ACE2. But they're not infected with a MERS coronavirus spike, which doesn't bind ACE2, right? It binds DPP4. So this is a very clear species-specific entry in these Pipistrellus ACE2 producing cells. So they say, we think based on all this that these are probably, that ACE2 is probably the natural receptor for these viruses in their natural hosts. And that's, the only thing missing is taking a Pipistrellus and removing ACE2, but you can't do that. There's no genetics, so not now anyway. Maybe one day. And then I like this. To confirm if Pipistrellus abramus ACE2 could function as a receptor for true virus. What do you think of that name, true virus, guys? Do you think, is it a true virus?
All the other ones were true. I feel like this virus is as true as the other ones were.
Well, except that you're playing on the pseudo and true.
Yeah, it's true. So they make an infectious clone, as Kathy said in the introduction, of HKU5, and they put green fluorescent protein in it. So
that's why I think it's not true.
Because of GFP.
Because that's GFP. Yeah.
I don't know. I would just, I see why they're calling it true. Because it's infectious. The pseudotype is infectious as well. So you can't just say infectious virus. True virus.
I think it just calls your attention to the fact that they're now doing a different experiment. And they're not doing pseudoviruses.
Okay. But I'm just trying to think if there's another way that I would say it. Would bona fide be good?
Well, Brienne's argument would still hold true if it's got GFP in it.
It's a bona fide virus.
Yeah, I mean, it doesn't have OR5 and it has GFP said.
Because bona fide means real or genuine.
Yeah.
And so pseudotypes are real or genuine. Okay, anyway, they make this infectious DNA copy and they get virus. And this virus can reproduce in either 293 or BHK cells that make the Pipistrellus abramus ACE2 without trypsin. So looks like the true virus binds to this and used it as a receptor to get into Pipistrellus, well, human cells. I guess it would get into Pipistrellus cells as well. That's the other thing you could do. You could get cells from Pipistrellus, and if maybe you could knock it out in those cells, right?
Yeah, maybe.
If they were a continuous cell line. I don't know. You could immortalize them, though.
Right. I guess the
Right.
question is, how hard is it to even, are these endangered or protected species where it would be hard to get cells?
Right. I also tried another cell type, these human liver cells, HUH7.5, that lack ACE2 and DPP4, and then they put receptors back into them. And so they knock out the receptors in these human cells, and then they put in the human, which restores susceptibility to SARS-CoV-2. They put in DPP4, which allows MERS-CoV to reproduce, and then they put in Pipistrellus ACE2, and the HKU5 GFP can reproduce in these cells as well. So multiple, pieces of evidence supporting this as a bona fide receptor, a true receptor, as opposed to a fake receptor. Okay, and then they start to look at some binding interactions here. They want to know if the HKU5 RBD recognizes the ACE2 of Pipistrellus. I think that it would have to do that to infect, but they're doing ELISAs. They see dose-dependent binding of the HKU RBD with Pipistrellus ACE2. I'm going to skip this one paragraph that I didn't understand at all. I don't understand what they were doing there, but anyway, we're going to now talk about structures. So there was a previous structure of clade 3 Merbecovirus RBDs from the PDF and neo-CoV viruses bound to ACE2 from Pipistrellus bats. That's very cool. So they wanted to know how the HKU5 ACE2 interface compares. So they use AlphaFold 3 to predict the interactions, and it's really good at it.
Mm-hmm.
Wow. Going to replace humans one of these days. So in the structure of PDF bound to ACE2, the interface involves multiple interactions between two surface exposed loops on the RBD, loop 1 and loop 2. And Alpha 3 said HKU5 is the same loops are going to be involved. But interestingly, the binding site on ACE2 was different from the PDF virus. Loop 2 is highly polymorphic, and Merbecos and several HKU5s have a 4 amino acid deletion in this region. So they want to know which amino acids are important for the interaction. So they do a bunch of mutagenesis assays on the RBD, and they find that changes in loop 2, but not loop 1 of the HKU5 RBD disrupt entry with Pipistrellus abramus ACE2. And interestingly, most of the changes reduced entry with bat ACE2, but changes in loop 2 of the HKU520S RBD, which is a different virus, but not HKU521S, increased entry with cells producing ACE2. So that's interesting.
It kind of sounds like this interface is undergoing quite a lot of evolutionary change to be able to potentially bind to lots of different versions of ACE2.
Yep. Which also made me think that it could change specificity as well pretty easily.
Exactly.
Okay. Now the last thing here that's interesting is they tried to solve the cryo-EM structure of a complex between HKU520S, the trimer with soluble Pipistrellus ACE2. All right. And all the structures they got were the spike only, no ACE2 density. So they derived the structure of the spike, 2.8 angstrom structure of the unbound HKU5 trimer, because they didn't see any ACE2 in the structure. And, it looks like other coronavirus spikes. There's an S1 and S2 subunit, which are the cleavage occurs between those two subunits. But so these RBD, the S1s, which contain the RBD in other coronaviruses, there are three of them, right? It's a trimer. Some of them are up and some of them are down. And the idea is that the RBD has to be up in order to engage the receptor. But in the down conformation, maybe it's good for immune evasion because a lot of antibodies are directed against the RBD. So they go up and down probably randomly. And when they're up, they can bind the receptor. But this structure is only down. So they don't know how, maybe they don't have the right conditions for them to go up because they have to go up to bind. But that's very interesting that they are down. That's kind of a subtle thing, right? A nuance, but I thought that was interesting. So there you go. The HKU5 binds the ACE2 from Pipistrellus. Let's see, what can we say here from the, they're talking about ACE2 and DPP4, not the receptor for clade4, more MERS-CoV, but maybe they have other receptors in other species that we didn't test, or maybe our test isn't good enough to pick it up. Couldn't be right. It is an artificial situation. The clade4s may bind to a different virus, which is analogous to Sarbeco-RBD clade2, which also can infect human cells through an unidentified route. So Sarbeco virus, not MERS-CoV, RBD clade2 viruses.
So we got to go back to the hedgehogs.
Go back to the hedgehogs, yeah. Okay. So, another thing that's very interesting. So there are other MERS-CoVs and Sarbeco's that the structures reveal the three S1s are all in the down configuration. And they're thinking, all the bad coronaviruses, it's the ones with these down configurations, they're gastrointestinal pathogens, not respiratory pathogens. And that's a very inhospitable environment, right? Low pH in the stomach, proteases in the gastrointestinal tract. They say a unique and harsh environment for viruses, which may select for the closed conformations in viruses that have this tropism. And how they do transition to the up, they don't know. That's a very interesting question there. Okay, so here we have synanthropic in the last paragraph. These bats are synanthropic, roosting in human dwellings and structures, so they can repeatedly infect people with the viruses in them. And, these clade2 MERS-CoV RBDs, they have low affinity for human ACE2, but you should probably still try to reduce contact, they say.
Yeah, and low is not no. Is not no,
but you can imagine that it could be selected for higher levels as well. So it's a nice study, shows you what you can do in cells and culture, and to really understand what level of threat. So here for HKU5, not a huge high level, but you still need to be doing surveillance and minimizing contact between humans and the host species. So very cool stuff. PDF. Okay. The Japanese house bat is also known as the Japanese Pipistrelle, is a kind of vesper bat. That's another word I really like, vesper. There's multiple meanings, right?
Mm-hmm.
Let's see. Dictionary, vesper. Vesper is a prayer.
Yeah, I was going to think it was something religious.
That's all it is. It's nothing else. Or it's a place. There are lots of vesper towns. There are people.
I think of vesper as being evening. Is that?
Yeah, evening. Actually, they say the archaic meaning is evening. It's an old meaning.
Apparently, it's also the name of a cocktail.
Interesting. Anyway, vesper bats, and they roost under the ceiling or inside the roof of old buildings. It's not just in Japan.
Right.
It's across East Asia, China to Taiwan, Korean Peninsula, and Japan. All right. That was fun. I enjoyed that very much. Let's do some 4:30. We could do could do some email. Kathy, you want to take that first one?
Sure. Joni writes, greetings from Albuquerque, New Mexico. The weather today is 65 Fahrenheit, 18 Celsius, going up to 81.27. Should be another beautiful day. It's been really nice for walks in the park. I've been catching up on my TWiV listening on those walks. A few weeks ago, you discussed Wendy Carlos and her iconic recording of Switched On Bach. I thought you said it was not available, but actually it is. Wendy had a major issue with her label over changing the attribution from her former (dead) name to her actual name for a long time. But about 25 years ago, she re-released the album with an additional track where you can hear her discussing the process of working out how to get the music to sound the way she wanted on the Moog. You can order her recordings, including the updated CD on her webpage, and Joni lists that. Also, I found a couple YouTube videos about Wendy and Switched On Bach. The first is apparently part of a 100-year anniversary release of material from Leonard Bernstein's Young People's Concerts. After having the New York Philharmonic perform the piece, he rolls out an actual Moog synthesizer and flicks some switches to have it play Wendy's version. And then there's the YouTube link for that. The second is a BBC interview from 1989 where you can see Wendy demonstrate how she recorded the music on the Moog. And there's a YouTube link for that. I especially will miss Dixon des Pommiers. I particularly enjoyed his own selections of the all-time jazz greats. I made a playlist on Spotify for the top 10 pianists, and I listen to it often. It's been a wonderful education listening to your program for 5 years now. Keep up the good work. All the best, Joni.
Yeah, I found my LP of my vinyl of Switched On Bach. I was very excited, and I really liked that. I didn't know that you could still buy it, so thank you for that.
Cool.
I also love when people say they've been listening for 5 years.
Mm-hmm.
I just think that means they like it, right?
Mm-hmm.
Brianne, can you take the next? Maybe you could take the next two, right?
Okay. Bill writes, Bill gives us a link to an Our World in Data page called Measles Vaccines Save Millions of Lives Each Year that came out in May 19th. And this shows a lot of the data on number of lives saved by measles vaccine. Great looking data, really well described here. And Bill notes that these are data RFK won't be spreading.
So the first graph is great, showing the lives saved by measles
that one's
compared
great. I love the next one too.
To others, but you can see how many lives all these vaccines have saved, right? That's very cool. Yeah, the next one is very good. The U.S. state level cases.
And where you can just sort of watch the number of cases go away in each state.
Yeah, I would like him to go through a 2025. That would be great, right?
Yeah.
Yeah, this is very good. Thank you for that, Bill.
And then Jason writes, hello, Twivanauts. It's longtime listener and episode 1000 attendee Jason here in New York City, where it's sunny and 20 degrees Celsius. I don't have a listener pick for once, but instead a quick story on how much you're educating the rest of us. A few weeks ago, my son was in the ER with what turned out to be norovirus. Fortunately, the Zofran worked quickly and he didn't have to stay overnight. While talking to the attending doctor, I asked a bunch of specific, detailed questions, prompting her to say, with all sincerity, "May I ask, are you a medical professional? You know a lot more about this than most people." I blushed, grinned under my mask, and replied that, no, I'm a computer engineer with a biochem degree, and all of my current knowledge comes from what I read and podcasts like TWiV. She laughed and said, that makes sense. Nicely done. Never doubt that the information and science you share make a difference. You're doing incredibly valuable work, and I am happy to be along for the ride. Jason.
Sometimes I do doubt it, but thank you. We appreciate these stories. Jay writes, thought you might like this article from the Sherbrooke Daily Record, published in Sherbrooke, Quebec, Canada in 1935, and is available through this website on page 37. Note you will need to turn translate on as the site is in French, without an offer to switch to English, which I don't speak, just a retired electrical engineer that learned a very little amount of German, Korean, Japanese, and Tagalog. The French I had in grade school is long forgotten. So he sent, Jay, is he or she, they sent a picture of a article from Saturday, February 23rd, 1935, in the Sherbrooke Daily Record entitled, "Science Has a Vital Role to Play in the World." In the subheading, "Entering Upon a New Area," Professor Julian Huxley declared last night at lecture at University of Bishop's College. And then you can sort of read it, yeah.
Mm-hmm.
Defending science against the claim that it has been responsible for the difficulties of the world during the past few years, Professor Huxley declared that science was entering upon a new area and would play an important role in the future welfare and prosperity of mankind. 1935. Cool.
Yeah.
Kathy?
Blue Pilgrim writes, a thing which should be understood about much of the West and the U.S. is that huge numbers of people have strong opinions based on belief and propaganda and ignoring actual evidence, and they refuse to talk about evidence. They don't change their opinions based on evidence because evidence was never the base of their opinions to start with. P.S. I also found this article. It's open access, so it's the one I read. And a Blue Pilgrim gives the link to that. T cells take up residence in the healthy brain via a gut-fat-brain axis. And the article
downloaded the PDF of that today.
Okay. And the article also mentions the research reported in this news article was supported by the National Institutes of Health awards 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 NIH awards and Yale University. So perhaps point this out to people and wonder if the cuts to NIH will stop this sort of research. It's inevitable that if you make these kinds of cuts, you will be stopping research. That's my comment. PPS from Blue Pilgrim. I also remembered a word I had forgotten, ethology, the study of behavior and sociology in non-human animals. And it turns out that MIT OpenCourseWare offers a free course in the subject, and one can also find some PDFs for free, including a few books. There's a Journal of Ethology, and gives a link to that, and also another link, an international society, and other resources, all for a word most people would not know, I guess, and which I almost did not. There's no N in ethology. It's not ethnology. Find out what your lab mice and backyard raccoons and pets are really thinking and saying to each other. I could guess they communicate as well as T cells. So thanks to Blue Pilgrim for a variety of interesting links and thoughts here.
Brianne.
Jinx writes, hello from sunny Tucson, Arizona. My name is Jinx Leoy. I'm a third-year family medicine resident, graduating in a month here, and I have been involved in a lot of global health. First, since this is my first email with you all, I want to thank you for the podcasts TWiV and TWiP. I've learned a lot from them and try to share that knowledge and the podcasts as often as I can. Recently, a colleague of mine in Sydney, Australia, published a collection of his essays about global health and research globally. It has more to do with the morality and ethics within healthcare and research around the world rather than the scientific experiments. However, the microbes that affect humans are global, and it's common to discuss papers from other countries. As scientists involved in clinically relevant topics, I cannot recommend this book enough. It's open access at the link below. It gives a link, and I've attached it as a PDF as well.
Foreign Gaze, the Essays on Global Health. Cool.
Awesome.
Congratulations on graduating.
Mm-hmm.
Yes.
And Heidi writes, I suppose this story will come across your radar, but just in case it doesn't, I know you mentioned Rocky Mountain Labs from time to time. My son used to have a desk there when he was working on a PhD at the University of Montana, and gives a link to a story, which I'll tell you about in a moment. Thanks, Heidi in Montana. P.S. I'm always appreciative of your outrage over all these destructive anti-science cuts and terrible policies. Don't let up. All right, so the link is an article, a news article in KFF Health News. And they say the title is Federal Cuts Ripple Through a Bioscience Hub in Rural Montana. So basically, some of the cuts are having an effect at Rocky Mountain Laboratories, which is a branch of NIH. And there's a picture here of some of the residents of Hamilton, a small town, who are often at odds with the lab because they don't want it to be there. They want to support the federal workers now, right? So they're picketing and showing signs. It's cool. Stop strangling science. So they're having problems at Rocky Mountain Lab as well, as you would expect. Oh, there's a picture of the entryway where I have gone in multiple times. In the background, the amazing mountains there. Cool. Thank you very much for that. Kathy.
Glenna writes, a cartoon you might enjoy. And it's a cartoon from the New Scientist that has some grammatical voices for use in scientific writing. Active voice, passive-aggressive voice, clickbait voice, haiku voice, and conspiracy voice. I have to admit that the only one I really read was the haiku voice. Quiet science lab. Workers arrive with samples. The testing begins.
Great haiku. I love that.
I like that. I think passive aggressive voice is kind of funny. We did all the collecting and testing. No need to thank us. Just doing our job.
Yeah.
Clickbait voice. We collected some samples. You won't believe what happened next. Anyway, you'll have this in the show notes, so you can look at the rest of them. They are really pretty funny. Brienne, you're next.
Sure. Brian writes, Dear TWiV, thank you for being there with scientific wisdom along with your passion for the scientific enterprise. This is especially relevant as the nation's sciences are brutally attacked. I'd like to share a letter I sent to the governors of the 7 Democratic-led states with the largest GDPs. They may not have political power, but they have fiscal power and need to use it. Dear Governor, you belong to a group that can save the world, the Magnificent Seven. You lead one of 7 Democratic states that together account for 38% of the U.S. GDP, the 3rd largest economy in the world. Of all of the Trump administration's destructive attacks, its impounding of 40%, $20 billion of NIH funding is the worst. NIH is the linchpin of U.S. scientific leadership. The Magnificent Seven can save critical scientific research by backfilling 38% of this lost funding. If each state in the Magnificent Seven increases taxes by 1.86% on incomes of at least $1 million, together that will account for the 38%. The administration's impoundment is illegal. States can claw back the funding they provide. This is important. Any recovered funding will be returned to the wealthy taxpayers. By acting in unison, states will make a difference. Their common action makes sacrifice shared. It mutes each state's right-wing attacks. By ensuring taxes will be returned if funds are restored, the wealthy will seek to have NIH funding restored. Each state has its own problems politicians want to increase funding for, but their constituents are concerned about national issues. They will understand a temporary tax paid by people with incomes of at least $1 million is for science across America. There are over 400,000 people with incomes of at least $1 million in the 7 states. That's well over half of all such people in America. All of the 7 can enact this tax, even California, where Democrats have the 2/3 majorities needed. Rise to the challenge and join in a Magnificent 7 states that demonstrate Democrats can act aggressively and imaginatively. Triage will be necessary, since 38% of funding is restored. But this is enough to maintain scientific momentum. Thank you for your dedicated service and thoughtful heroism. Sincerely, etc. Perhaps others can encourage these states to take scientific leadership.
Wow. That's a real interesting approach.
Yeah.
I think most of these people with that income are Republicans.
So if you tax them and then say you'll get your money back if you get rid of these cuts.
Yeah,
They might,
I think it's
they might
I like
fight
the
for it.
idea.
Yeah.
I like the idea. I think it's very good. I just doubt that, California could do it, but I don't know about the other states. I wonder what the other states are. That would be interesting.
I was
to
trying
know
to figure that out myself.
if they're red or blue, right? That's going to make a difference.
Well, he said they all have Democratic-led, so governors of 7 Democratic-led states.
Yeah. Interesting. It would be interesting if a lot of people sent in this letter.
Mm-hmm.
So yes, New Jersey has a Democratic governor,
Right.
and so does New York, right?
Mm-hmm.
So the first thing I find is the 7 states with the highest GDP, but they're not all led by Democratic governors. So It's a list of 7, but
Yeah.
not enough.
Well, thank you, Brian. I'm going to try sending that to Phil Murphy. All right. Time for some picks of the week. Brianne, what do you have for us?
I have a story that I came across in a couple of different places. I've linked the CNN article. It's for the cat lovers of TWiV. And this is an article about orange cats, and a paper that just came out recently that describes a mutation that was found that allows orange cats to be orange. Which is really interesting. The researchers had sort of known about, of course, orange cats for a long time. They'd known that most of them were male, and so they assumed that this was going to be a mutation on the X chromosome, but they'd never found it before. So they were able to take samples from a large number of orange cats and start looking at their genetics, and they found that there was actually a non-coding change in a gene that is not normally even expressed in melanocytes. But this change led to ectopic expression of the gene in the melanocytes and blocked the production of black pigment, thus making the cats orange. And sort of this had never been seen in any other organisms. So I thought that was particularly interesting and something that, one of many things we can learn from our cats. I also thought it was interesting that they were thinking that they might see something similar in tigers and some of the big orange cats, and they do not. And so there's still work to be done here.
That's neat. You have an orange cat, Brianne?
I do not. My cat is gray.
That's neat.
It is.
We talked about orange cats on TWiV a couple of months ago. It's a different mutation, though. A different cause of orangeness.
Yeah, but
Kathy,
I liked that it wasn't a coding mutation.
Yes, that's very cool. Kathy, what do you have for us?
I have something that I've had in my queue for a while. It's a middle schooler from Chicago had a STEM project, and they were supposed to go out to nature and collect samples, and some of them collected leaves and sticks and other stuff. And this particular person collected goose poop. And having just been at Ann Arbor Park this last weekend, where there was a lot of Canada goose poop around, it kind of reminded me of this. Anyway, from this goose poop, they had a project to identify bacteria with antibiotic activity, and her goose poop sample had that. But then the researchers continued with that goose poop sample, and that led to an anti-cancer compound. So I just thought it was a cool story about encouraging science in young people, and it's just a nice, inspiring article about the project and the interview with the student.
That's very cool.
That is really cool.
This student will likely be a scientist because of this cool project, or maybe a doctor.
Yeah.
All right, I have another Times article, which is probably paywalled, but it's really good. It's an opinion piece by Michelle Goldberg. It's called "Elon Musk's Legacy is Disease, Starvation, and Death." And it starts with this wonderful paragraph. There is an Elon Musk post on X that should define his legacy. We spent the weekend feeding USAID into the wood chipper, he wrote. He said he could have gone to some great parties, did that instead. And she goes on to write, his absurd scheme to save the government $1 trillion has been a failure. But there's one place where he achieved his goals. He did indeed shred USAID, terminated more than 80% of USAID grants. Brooke Nichols, an associate professor of global health at Boston University, has estimated that these cuts have already resulted in about 300,000 deaths, most of them children. That is what Musk's foray into politics accomplished. And of course, the White House officials deny all of that. Atul Gawande, we've heard of him many times here on TWiV, told me that during a trip to Kenya, He visited the National Referral Hospital. There's been a major increase in the number of patients with advanced HIV as a result of losing access to antiretroviral medication at the refugee camps on the border of South Sudan. Food aid has been cut so severely that people are getting less than 30% of the calories they need. It's not enough to survive on, and that has caused skyrocketing levels of severe malnutrition and deaths associated with it, according to Gawande. And it's a good opinion piece, which has more about Musk and all of this stuff. So I just think that this is so mean. It's unbelievable.
I know. I just can't understand it.
Well, because he has no idea what's going on. Or maybe he does, and he's just a mean person. I don't know. Michelle Obama said, being president doesn't change who you are. It reveals who you are. We have a listener pick from David. Thank you for your continued efforts to support science and to counter the rampant misinformation around us. I submit for your consideration as a listener pick a book from Carl Sagan in 1995, The Demon-Haunted World, Science as a Candle in the Dark. And Wikipedia has a summary of the book. The subjects that Dr. Sagan covers are eerily relevant to events today. The solace I take from his coverage of similar past events is that we eventually emerged from those past dark periods, at least for a time, recovered to some extent. Whether that applies to current times and how long the recovery will take, if it does occur, remain to be seen.
It's a great book.
You've read it?
I have, although I read it in high school, so it's been a while.
I'd like to read it. My question, perhaps best put to Dr. Griffin, well, he's not here, so let's try, is hopefully purely hypothetical. If some series of events were to remove the ability of U.S. citizens to obtain needed vaccines or needed medical care, where in the world would Dr. Griffin recommend patients consider traveling to for care? Canada comes to mind, but I'm not sure how welcome U.S. citizens would be and whether Canada could support an influx of medical tourists. Thanks for all that you do. It's interesting. I asked Daniel this last night in a kind of slightly different way on the clinical update. I said, what percent of let's say that vaccines are no longer required anywhere in the U.S. What percentage of Americans do you think would decide not to vaccinate themselves or their children? And he said, I don't remember the number, 30%. It's a lot of people, many millions of people. And that would cause an increase in many infectious diseases eventually, maybe not immediately. We did a paper on that on TWiV a couple of weeks ago, how long it would take. But, the rest of the country would like to be vaccinated, and hopefully they can still do so in the U.S. But if not, I don't know where you could go. I mean, there are many European countries you could go to as well, but why would they want to vaccinate Americans, right? I think that's a good question for Daniel. I will put it to him next.
Well, they might
week.
want to vaccinate Americans because the viruses are not going to stay in one place.
Yeah, but no one is safe till everyone is safe.
Exactly.
That's right. That's a good question, David. I like that very much. Oh my gosh, it's so upsetting. And it didn't have to be this way. Just very upsetting at many levels, but science and public health, it is just, this idea that RFK can just say what vaccines you can have now or not. It's ridiculous.
So ridiculous.
And especially since the vaccines, he doesn't want anybody under 65 to have a COVID vaccine, a booster anyway. Who knows? It's not even clear. But there's no data saying that they're bad.
No.
He thinks they're terrible. And, the thing is that they've been licensed for that age group already. So he's going to take away, theoretically, the license.
Yeah.
And there's no data, and Bhattacharya and Makari, they say, oh, this is data-based. Where are the data that say it's dangerous to vaccinate? But there's no data.
No, and if they show us data, I would like the data to be data that has a real citation.
That's just what I was thinking.
Yes, there should be a paper. All right, that is TWiV 1223. You can find the show notes at microbe.tv/TWiV. Questions, comments, picks of the week, TWiV at microbe.tv. And once again, we'd love your support to continue these programs of real science. Go to microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker is at Drew University, bioprof, barker.bsky.social on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv, on Blue Sky, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.